Hello students, so today in this video we will discuss about enzymes. What are enzymes, their structure and function, what are the chemical reactions they help in, that is metabolic reactions including catabolic and anabolic reactions, and at last lock and key hypothesis and the induced fit hypothesis, that are the two well-known hypotheses of enzymes. Okay, so before directly jumping into enzymes, let us look at something else to understand enzymes. So, do you know what our bodies are made up of? All the living organisms, including humans, are made up of cells. Now, there are trillions of cells present in a human body, and all these cells undergoes several metabolic reactions, including anabolic reactions and catabolic reactions. But before that, do you know what is a metabolic reaction? What is a reaction, first of all? Let us see that. The reactant A and B forming a product C. We call this reactant either as a reactant, or we can also call it as substrate. The chemical reaction has taken place and we have a product. But what is the problem here? The problem is this reaction that we have seen is extremely slow, and because of this slow reaction our body cannot cope up. That is why we need something that elevates or increases speed. But do you know what can increase the speed of a reaction? Something that is called as catalyst. Yes, what increases speed of the reaction? A catalyst. And when we are talking about a biological reaction, we can also call this catalyst as a biocatalyst. Biocatalyst meaning, bio means something that is living and catalyst is something that increases or enhances the speed of any reaction. But why are we talking about this catalyst? Because this catalyst is the enzyme. Yes, this catalyst is also called enzyme. Okay, so now what is an enzyme? Enzyme is a biocatalyst that increases the speed of reaction without being used up, and it can be used over and over again. Yes, see. First of all, you are supposed to know that an enzyme is made by several amino acids joined together in specific shape. Yes, now you would say that when amino acids are joined together, they form proteins. Why am I calling it enzyme? Because enzyme is also protein. Okay, so now we know that enzyme is something that increases the rate of reaction. But do you know, without the help of an enzyme, if a reaction takes place, it would take days, maybe months, or even years. With the help of enzymes, the same reaction will be carried out 100 times faster. Before going into the structure of enzyme, do you know enzymes are very specific and all the enzymes have very different and unique shapes? Okay, so now you got to know what is enzyme and how it works. But let us see the structure of enzyme. Okay, so now considering this as a structure of an enzyme, every enzyme has got a specific region. Only in this region the substrate is allowed to come and bind. So this is the hollow region where substrate can come and bind, and this is called as active site. This is our enzyme. Okay, so now we have taken the enzyme. Now what do we need to carry on the reaction? As we discussed earlier, A plus B. Reactant A or the substrate A plus reactant B or the substrate B gives the product C. Okay, so when it gives the product C, when there is the presence of enzyme. So we need enzyme here. But the thing is, It does not mean that the reaction will not go on if there is no enzyme. Reaction will still go on if there is no enzyme present, but the time taken will be 100 times. So it will be extremely slow. Alright, so we have an enzyme with active site. Now we need substrate. What should be the shape of substrate? It should be extremely complementary to the active site. So if this is the active site, the shape should be somewhat like this. Yes. Is it 100% complementary? Yes. Only in that case, reaction will go on. Okay, so this is the shape of the substrate. Now, what happens further? It forms ES complex. This is the enzyme. Enzyme plus substrate makes enzyme substrate complex. So here, okay, so here as you can see, enzyme and the substrate are combined to each other very tightly and they form ES complex, that is enzyme substrate complex. This happens for a very short time. Okay, so ES complex is formed. Further, we have the product. So as we know, the ES complex has been formed for a very short period of time and quickly it gives us the product. This is the enzyme in very same shape as it was in the beginning. The enzyme is free now and we have got different products, 1 and 2. Earlier it was a whole single substrate. Now this substrate has divided or broken up into 2 parts and this is our product. So from this, can we say that the enzyme is not being used up in the reaction? Yes, enzyme is not being used up in the reaction and it can be used over and over again, but only when it is very specific to its substrate. And what decides the specificity? Its shape. So basically, if you see an enzyme, it is nothing else but enzyme is the linkage of different amino acids in a specific and unique shape. This shape is what decides the specificity of the enzyme to its substrate. Okay, so now there is one question. Which reaction is this? Catabolic reaction or anabolic reaction? If you want to answer this, you have to see the substrate. This large molecule of substrate is broken up into 2 different parts. Means that larger molecule is broken up into smaller molecules. The breakage of the molecule is called catabolic reaction. And the vice versa is anabolic reaction. So what happens in anabolic reaction? This substrate would be like this. 2 different substrate combining this way, 2 different, and then finally forming a single product. Do you understand the difference? As you can see, this is catabolic reaction. In catabolic reaction, large molecules differentiates or breaks up into different small molecules. Vice versa happens in anabolic reactions. In anabolic reactions, different small molecules combine together and form a single large molecules. Here, energy is released. And here, they need energy. Okay. Okay, so far we looked into what is enzyme and its structure, but now we are supposed to look at two hypotheses of enzyme. So the first one is lock and key hypothesis. Now what is that? As any specific lock has got its own key, so you cannot open that lock with any random key. Similarly, here lock is our enzyme having this active site. Okay, so now this is our enzyme. Enzyme will carry on the reaction only when it gets its own substrate, which is 100% complementary to the active site of enzyme. What do I mean by complementary? This enzyme should get a substrate that fits perfectly inside this enzyme, like this. As you can see, it fits perfectly in this active site. Yes, it does. So in this case, the first one is called lock and key hypothesis. So as you can see that in this enzyme, it has got an active site and this substrate is very specific to the active site. What if I would have a different substrate? Something like this. This is a substrate. Will this substrate fit into this active site? No, it won't. If we provide this substrate, reaction will not further go on and we will not have the product. So, lock and key hypothesis says that the substrate should be exactly complementary to the shape of the active site. The rigid enzyme shape never changes. No flexibility is seen in the enzyme. Yes, we saw there is no flexibility. This enzyme could not work with this substrate. It needed something that is very same to the shape of the active site. And third and the last point, enzyme and the substrate should be 100% complementary to each other. So now we learned what is lock and key hypothesis. Now let us focus on induced fit hypothesis. Lock and key was a traditional method which was accepted long back. But now there are some modifications into lock and key method and this is induced fit. According to the induced fit method, if this is the enzyme, we do not need a complementary shape. 100% complementary shape is not required. First point, 100% complementary enzyme and substrate are not required. If the enzyme has an active site which is little different from the substrate, let's say this is our substrate. The substrate should not bind to the enzyme according to the lock and key hypothesis. But here, induced fit hypothesis says that enzyme has a capacity to change slightly only when the substrate comes and binds to it. So as you can see, the enzyme has an active site which is different from the substrate. Still, it can bind and form the ES complex. Further, they carry on the reaction and form the product and gives up the enzyme as it is. So what you have to note here is, see, the shape of the enzyme is same as it was in the previous stage. The shape of enzyme changes for a very little amount of time and then goes back to the initial stage. This means that enzyme does not get consumed in the reaction, neither it is used nor it changes its shape before or after the reaction. Now we get the product. So this is the enzyme and this is the product. So, according to this hypothesis, what are the different things happening than lock and key hypothesis? First one is 100% complementary enzyme and substrate are not required according to this hypothesis. Secondly, enzyme is not rigid, it is flexible. Enzyme and the substrate fits each other the way your glove and your hand fits each other. So they both are flexible to each other and they forms a tight structure. Okay, so I hope you understood this topic. That's it for the video. Thank you.